How much like mushiness and mess is there? Because taking biology classes, the diagrams are always really clean and crisp. Neuroscience, the pictures of neurons are always really nice and very. But whenever I look at pictures of like real brains, they're all, I don't know what is going on.
Yeah.
So how much are biological systems in reality? Like, how hard is it to figure out what's going on?
Not too bad. Once you really get used to this, that's where experience and skill and education really come into play. Is if you stare at 1,000 brains, it becomes easier to kind of mentally peel back the, say, for instance, blood vessels that are obscuring the sulci and gyri, kind of the wrinkle pattern of the surface of the brain. Occasionally, when you're first starting to do this and you open the skull, it doesn't match what you thought you were going to see based on the MRI. And with more experience, you learn to kind of peel back that layer of blood vessels and see the underlying pattern of wrinkles in the brain and use that as a landmark for where you are.
The wrinkles are a landmark? So like...
Yeah. So I was describing hand knob earlier. That's a pattern of the wrinkles in the brain. It's sort of this sort of Greek letter omega shaped area of the brain.
So you could recognize the hand knob area? Like if I show you 1,000 brains and give you like 1 minute with each, you'd be like, yep, that's that.
Sure.
And so there is some uniqueness to that area of the brain, like in terms of the geometry, the topology of the thing.
Yeah.
Where is it about in the...
It's so you have this strip of brain running down the top called the primary motor area. And I'm sure you've seen this picture of the homunculus laid over the surface of the brain, the weird little guy with huge lips and giant hands. That guy sort of lays with his legs up at the top of the brain and face, arm areas farther down, and then some kind of mouth, lip, tongue areas farther down. And so the hand is right in there, and then the areas that control speech, at least on the left side of the brain, in most people are just below that. And so any muscle that you voluntarily move in your body, the vast majority of that references that strip, or those intentions come from that strip of brain, and the wrinkle for hand knob is right in the middle of that.
And vision is back here?
Yep.
Also close to the surface?
Vision's a little deeper. And so, this gets to your question about how deep can you get. To do vision, we can't just do the surface of the brain. We have to be able to go in, not as deep as we'd have to go for DBS, but maybe a centimeter deeper than we're used to for hand insertions. And so that's, work in progress. That's a new set of challenges to overcome.
By the way, you mentioned the Utah array, and I just saw a picture of that, and that thing looks terrifying.
Yeah,
because it's because it's rigid, and then if you look at the threads, they're flexible. What can you say that's interesting to you about that kind of approach of the flexible threads to deliver the electrodes next to the neurons?
Yeah, I mean, the goal there comes from experience. I mean, we stand on the shoulders of people that made Utah arrays and used Utah arrays for decades before we ever even came along. Neuralink arose partly, this approach to technology arose out of a need recognized after Utah arrays would fail routinely because the rigid electrodes, those spikes that are literally hammered using an air hammer into the brain, those spikes generate a bad immune response that encapsulates the electrode spikes in scar tissue, essentially. And so one of the projects that was being worked on in the Anderson lab at Caltech when I got there was to see if you could use chemotherapy to prevent the formation of scar. It's like, things are pretty bad when you're jamming a bed of nails into the brain and then treating that with chemotherapy to try to prevent scar tissue. It's like, maybe we've gotten off track here, guys. Maybe there's a fundamental redesign necessary. And so Neuralink's approach of using highly flexible, tiny electrodes avoids a lot of the bleeding, avoids a lot of the immune response that ends up happening when rigid electrodes are pounded into the brain. And so what we see is our electrode longevity and functionality and the health of the brain tissue immediately surrounding the electrode is excellent. I mean, it goes on for years now in our animal models.
What do most people not understand about the biology of the brain? We'll mention the vasculature. That's really interesting.
I think the most interesting, maybe underappreciated fact is that it really does control almost everything. I mean, I don't know, for an out of the blue example, imagine you want a lever on fertility. You want to be able to turn fertility on and off. I mean, there are legitimate targets in the brain itself to modulate fertility. Say, blood pressure. You want to modulate blood pressure. There are legitimate targets in the brain for doing that. Things that aren't immediately obvious as brain problems are potentially solvable in the brain. And so I think it's an underexplored area for primary treatments of all the things that bother people.
That's a really fascinating way to look at it. Like, there's a lot of conditions we might think have nothing to do with the brain, but they might just be symptoms of something that actually started in the brain. The actual source of the problem, the primary source is something in the brain.
Yeah, not always. I mean, their kidney disease is real, but there are levers you can pull in the brain that affect all of these systems.
There's knobs.
Yeah.
On-off switches and knobs in the brain, from which this all originates.
Yeah.